With CRISPR and CRISPR screens being so powerful, there are many computational challenges involved for both CRISPR editing and screening. I want to list in here a whole list of CRISPR computational resources. You can click on this and see many computational biologists are developing resources to help CRISPR experiment. And I just kind of want to show you what people think about in terms of CRISPR. The first is CRISPR guide design. So we remember, in order to knock out a particular gene, you need to identify a unique piece of DNA followed by NGG, or in this case, it's Cas9. The PAM sequence is NGG. And so as long as you design, the sequence before can be 19, 20, or 21 nucleotide long before the NGG. And so I have a quiz for the students in here. How often do you see an NGG sequence in the genome on average? Like if I want to knock out the gene, how often can I design a guide RNA sequence? Can you guess? Oh, very good. That's an excellent answer. Because the first G happens on average every 4 base pairs, right? ACGT. The second G happens also on average every 4 base pair. And so you have 2 consecutive G, that's 1 in 16s, but this can happen both on the plus strand and negative strand, so that doubles. And so, yeah, on average, it's every 8 base pair. You will see it either on the plus strand or the minus strand, because either strand can bring the Cas9 to the location, and because it makes the double strand cut. And so, of course, in the genome, the CG sequence is not exactly half. In human genome, you probably have more AT than GC. So I would say on average, NGG happens roughly every 10 base pair, which means if you have a gene that's thousands of nucleotide long, there are hundreds of guide RNA that you can design. But then turns out they don't all work equally well. If you want to knock out the gene, for a scientist to do this experiment, it takes a lot of effort. You want to make sure you design a guide and it will just work well and really knock out the gene, right? So what are the design rules? How come some works better and others don't work so well? So yeah, we mentioned guide RNA have varying efficiency. Maybe it cannot express highly, or maybe it has a secondary structure which prevent it from bringing the Cas9 to the location, or for some reason the Cas does not cut well, or maybe the repair is not done right or something. They just have different efficiency. And so one approach that our group have used to design guide RNA is to say, well, there are housekeeping genes. If you look at many CRISPR screens in cancer cells, these genes are always essential, such as ribosomal genes. Clearly this gene is important. You knock out the gene, the cell will die. And we know that these genes are important. And so in the CRISPR screen, do you expect them to be positively selected or negatively selected? Do you expect their guide RNA to have a positive fold change or negative fold change if you grow the cell after 2 weeks' time? Could we ask the students this? Yeah, it's negative. Right. The essential genes or the housekeeping genes are supposed to be negatively selected, meaning that if you grow the cell over 2 to 3 weeks' time, their guide RNA population or fraction should decrease over time. So you expect a negative fold change compared to the initial condition. And so if we take all of those genes that are clearly housekeeping, we also call them pan-lethal genes, meaning that in whatever experiment they are essential. And we look at all their guides. And this is the CRISPR screen in one cell line. This is the CRISPR screen in another cell line. And these are all the guides targeting, say, ribosomal genes. They are pan-lethal. And you see some of these guides, they give you a negative fold change in both cell lines. We would call them as efficient guide. They work very well. You get the desired phenotype. But these other guide in here, they target an essential gene, but they really don't give you the desired phenotype. You don't see a negative selection. And the red dots here are control guide which don't even appear any hits in the genome. It's not targeting any location in the genome, and they give you kind of a zero-fold change or like no change. And so we wonder, what is the difference between these guide RNA that work well from those that don't work so well? And we could look at the sequences, and remember, each guide sequence is about 20 nucleotide long. You can take the 20 nucleotide sequence as well as the 10 base pair before and after the sequence, and we can encode them into a string of, A is 0, C is 1, G is 2, and T is, sorry, A is 0, C is 1, G is 2, and T is 3. And you encode this is kind of a binary encoding, one-hot encoding, sorry. And so you can encode the original sequence into a string of numbers. And those guide that target essential gene and give you a negative fold change, you call them as working, efficient ones, and we give them a code of 1. But those that target essential gene but don't give you the desired phenotype, you say, okay, these are not working, it's -1, or you can be 0. And you can develop machine learning algorithms to really see, what's the difference between those working one and not working one in terms of sequence. And indeed, we could build those features, and we can see that this kind of goes back to your previous machine learning methods. Regression-based approach, you can use random forest, you can use many other supervector machines, to try to predict the efficiency. And this is the sequence model that we built. This is the particular PAM sequence, NGG. And if a particular sequence, in terms of a motif match, look more like the sequence above, it's gonna be a more efficient guide. But if it has a lot of these sequence in below, it won't be an inefficient guide. And so we could use this to design CRISPR guide that will really, go to the right location and really make the cut, and you will get the desired phenotype. And we can use it to design guide across the whole genome. So that's kind of our approach. There's another study where in order to target a particular gene, these are not introns and exons. These are all exon sequences, but there are sequence domains, because on the proteins, there are certain functional domains, but there are also spacers to connect them together. And so this is Chris Vacock's group. They design guide across the genome, across this whole gene, because you can design, hundreds of different guide around here, and they want to see which guide really give them the correct phenotype. And this is one gene, this is another gene, and they see that when the guide is targeting these sequences that are not in the functional domains, they don't really create a strong phenotype. By the way, these two genes are both important genes. And so, but then if the guide is targeting the more important functional domains, it will create a stronger phenotype, which is kind of, it makes sense, because sometimes when you make a cut, when it repairs, sometimes the repair could be in frame, meaning that you just add one full amino acid or remove one amino acid. But if this location doesn't matter as much, for example, in the proteins, there are certain secondary structures that are important. But then there are also spacers, which are protein loops. If you miss or add one amino acid, it doesn't matter. Those usually don't create a strong phenotype. Whereas on the domain, say on the double helix, you suddenly change, you add or remove one amino acid, the whole structure of the protein would change. It will not be very happy. Then you will see the strong phenotype. And so from this, what we learn is that understanding the protein domain is important. And if you want to use a proxy, you can use evolutionary conservation, because if you look at the protein sequence or genome track, a lot of these domain sequences have very strong sequence conservation, whereas it's not so essential regions, the conservation would be lower. And so if we have the sequence conservation and also the sequence preference from here, you can usually design guide RNA that will give you very good efficiency. Okay, so that's on the guide or the efficiency part. It's also important to make sure that the guide you design does not have off-target effect. This is especially important if you want to target some locations to really repair the genome, say a genetic disease, right? You don't want this to go everywhere else to cut and repair and create undesirable changes into the child. You correct one mistake, but created a lot more mistakes. That would be really bad. And so there are computational approaches. You first need to make sure the guide RNA really only match to a unique location onto the genome. But we also look at where you could have off target. And so again, we look at many genes where each row is one gene, and we design many different guides along the gene. You can see most of the genes give you similar selection level, but then we would see some genes that suddenly give you very strong outliers, either significantly positively selected or significantly negatively selected, which suggests that something is wrong with this guide. It's probably giving you an off-target cutting effect. And so when we look at those sequences that are outliers compared to the rest of the guide RNA on the same gene, we notice something as well. Remember previously from here, we can see that guide RNA that have more Gs are very, very efficient. Turns out they can also give you very bad off-target effect, which are these outliers. Remember, there are regions in the genome that are like CPG islands, so they're very CG rich. And so what we are suspecting is that if you have a guide that have a lot of CG, in addition to going to the correct target, it probably kind of have a little bit of target binding to some CPG rich regions. Even though it can go there and the cutting is not, when the match is not perfect, it doesn't really cut efficiently. But because there are so many CG regions in the genome, if it goes to hundreds of CPG islands and each location make a small percentage of cut, you still create a lot of cuts in the genome, which is harmful for the cell. And so in some sense, you want to create the correct sequence that have some kind of correct or like might have some favoring of G positions, but you don't want to have too many Gs in the guide, which might create off-target cutting. Sometimes scientists want to know, if I introduce a particular cut, how does the repair work, right? And so depending on whether the repair is non-homologous end joining, meaning that we just make the cut, no template, and you just see where, how does the cell repair. The other one is the homologous recombination, which use the template you provide to repair the cut. And so in this approach, usually after the cut, scientists will use PCR to amplify out this region, followed by high throughput sequencing, and really read out each sequence to see exactly where the cut happens, how does the repair happens. And so this is just looking at, where is the kind of the PAM sequence, where does the cut happen and what is the repair? What is it, insertions or deletions, exactly what happens, and whether this creates a frame shift mutation or in-frame mutation. And also looking at, how much insertion happen, or it gets, because the insertion can also be reverted and so on. And so these are algorithms that can tell you what is the exact repair. It's again, just this is a single cut at a time, but you PCR amplify this region out from all the different cells that carry this guide RNA for the genome editing, and you read out the sequence high throughput to understand the actual repair result in that cell and understand whether that gene get knocked out. Another important analysis is to analyze the CRISPR screen for hits. So let's look at this situation. Remember, in each gene, in order to make sure that on this gene we are creating the correct knockout, sometimes you use multiple photos. In one cell, you use this guide, another cell, you use the same, different guide for a same gene. So this is the gene 1 guide 2, this is gene 1 guide 3, and so on. a lot of CRISPR screens use 10 guide per gene, just to make sure that if one guide doesn't work so well, you still have the remaining one that work well. And so this is another gene, you have different guides. And supposedly in the beginning of the experiment, we try to have an even library. We infect the cell and you do the sequencing in the day 0, right after infection. You say, okay, after infection, show me where your photos. And you look at the guide sequence abundance. Okay, you can see they're roughly even around 100. And then you grow the cell for 3 weeks time, and you take out the cell again, let the cell kind of PCR amplify out the guide RNA sequence, and then look at their abundance. You could see in this case, right, this is a negative selection. The guide is becoming depleted. This means this guide is, sorry, I don't remember what this number is. It should be 1/10. Yeah, I can maybe, sorry, let me correct this. Yeah, okay, 1/10. Yeah, so the fold change is kind of, well, yeah, 1/10. This means the cell is negative, the gene is negatively selected, the gene is growth essential. But in another case, we see a 10-fold increase. This is growth inhibitory. Of course, we are looking at the most ideal situation. The reality is, even in day 0, you don't really see this very even situation. There are already some distribution, and after 10, 20, 21, in this case, you can see the 4 guides are giving you very consistent performance, but sometimes they don't. And so it can get pretty complicated. The question is, how do you normalize the raw data and compare the 2 conditions? So this is kind of now thinking about, sequencing. You have 2 libraries, you have to normalize so that overall, by read abundance, you need to kind of normalize them together. Most of the genes in the CRISPR screens will not have a phenotype at all. They should be close to 0, like the day 0 and day 21 should be quite similar. A portion of the genes will be negatively selected. Some genes will be positively selected. So I have a quiz question for the group. Would we see more genes negatively selected or more genes positively selected? Would we see more genes negatively selected or positively selected? So to answer that question, let me ask another question. If you have a car and the car has many different parts, and we ask a person to go there and just get rid of a part, and we ask how many parts will make the car run slower, and how many parts you just randomly take them out will actually make the car run faster? Would you see more parts that take out will make the car run slower, or more parts that will you take it out? Yes, most of the time when you take out a part, it's gonna make the cell slower. Or maybe it doesn't matter, you take out the seat, it's not gonna change too much, right? And so in this situation, vast majority of the genes will not have a change. So you can use those to normalize to make sure the 2 libraries are about, some more comparable. There will be some genes, usually, like 20, 30%, or maybe 10% of the genes that will make the cell grow slower, but very few genes. We're talking about, like a few hundred genes will make the cell, you knock out the gene, the cell will grow faster. It's kind of a much rarer event. And so how do we really analyze the data? In the class homework, we're going to ask you to try out one algorithm that we developed, which have been widely used. It's called MAGECK. The idea is like this. When we have each guide, after you normalize the overall data so that most of the guide in day 0 and 21 are actually similar, they have no fold change, we rank all the genes by their fold change between the 2 conditions. And then we take all the genes, and so you can see some genes, this is from a most negative fold change to, or like a log fold change. to a most positive log fold change, and you rank all the genes by their p-value of differential abundance. And then we look at all the guides that target one particular gene. If this gene doesn't really have a phenotype, their guides probably are randomly scattered, and especially towards the zero, meaning that there's no fold change, no log fold change. Its log fold change would be close to zero. But if there is a guide that is important in here, well, sorry, if the gene actually have a phenotype, we will see in this case, a lot of the guides are skewed to either the left or skewed toward the right. In this case, these guides probably don't work very well. This is in the early days of CRISPR screen, but these guides work very well. It gives you a negative fold change. This is an essential gene. And so we are seeing, the guides are skewing to the left. And so we can calculate how much the guides are skewing to the left or to the right to give us a p-value significance about whether this gene is essential or not. In order to do this, previously we have, earlier in the course, we've done an algorithm, or we've shown you an algorithm that looks like this type of skew, either to the left or to the right. Do you guys remember what that statistical test is? And where did you use it? You remember? Yes, KS test for gene set enrichment analysis. It's perfect. Yeah, so remember in that, we are ranking all the genes from the most negatively expressed downregulated gene to the most upregulated, or from the most upregulated gene to the most downregulated genes. And then we look at each pathway, each gene ontology, for all the genes that are annotated as having this pathway or gene ontology annotation, and we ask whether those genes are overall skewed to the downregulation or upregulation or more to the downregulation. And using that, we can see whether, overall you see a signature or pathway upregulated or downregulated. We can use something like that. In this case, we're using kind of a non-parametric order statistics to give us a p-value. And in the homework, you will be using some of that to look at a CRISPR screen. Later on, we developed another algorithm. It's when you have many different CRISPR screens. And so you have one gene that have, say, in this case, 7 different guides, and we might have many conditions, right? So you can treat the cell with one drug, or maybe this guide in many different different cells. So you can do this in first cell, second cell, third cell, and so on. You look at many different CRISPR conditions using the same CRISPR library to do the knockout. And we can look at them together to see which guide actually works better. You can notice that some of these guides don't work very well. Other guides might be working well. And so we are trying to, first of all, look at the guide RNA efficiency. There's also sequencing over dispersion. This is similar to RNA-seq. You see sequencing all having over dispersion. And we also want to look at differential expression or differential abundance of the guide on all the on this gene across all the guides together. And so the approach we're using is an expectation maximization approach. You can imagine like this. Initially, we have 7 guides from the design. We can use our algorithm to, based on the sequence and conservation, to say, okay, roughly these are, this is a good one, this is not so great. Or if you don't even know, you can just say, okay, initially all the guides are equally good. This is like students coming to Harvard. We use their GPAs, use their SAT scores to roughly decide, okay, these are good students, these are mediocre, these are probably not so great students. And then we look at how they supposedly the students are taking the same course. We're trying to decide, whether a professor is teaching well, meaning whether this gene is being selected, positively selected or negatively selected in a CRISPR screen. At the same time, we try to also decide whether the student is a good student. So how do we do that? If you assume all the guides are equally efficient, based on this, initially you can say, okay, based on their selection, let me just take an average. You take an average, you decide, okay, this is a good professor, this is not so great a professor, this is terrible, and this is pretty good. And so you look at all the courses they've taken together. And based on this, you assume that if this is a good student, its ranking of the professors should be very good. So if the guide actually agrees with the gene overall across the different cell lines, that student is good because it's able to rank the professors correctly. Whereas some bad students, it's like crazy, right? They're just really no correlation with the professor's actual performance. You will downweight them. So the efficient guide are good students. And the next time when you look at the professor's course evaluation, you give the good students a little higher weight and the bad students a little lower weight. And then you rank the professors again. And based on the professor's ranking across different courses, you reevaluate your guide and you ask, which student is ranking the professors well? And so we do this iteratively. After only a few rounds, we could usually finish, the program will converge. And at that time, we will know how much a particular gene is being selected compared to the zero condition, to the initial condition, and which of those guide sequence are efficient guide versus inefficient guide. And so this is also kind of a widely used CRISPR screen algorithm. So this is like we look at the gene selection, which is rank professors, and next one is the read count. We evaluate all the guide. The E step is trying to see which of this guide, the performance across this guide in all the conditions agree well with the gene. And so iteratively, you will get a better result. Okay. And so these are some of the computational algorithm we mentioned, and I would revert you to the very first slide on all the tools that people are using in CRISPR data analysis.